Hi everyone. We're just going to go through some problems. These are the same problems we went through Wednesday during class 4. But just so you have a video of them in case that's helpful. So our first question is, what is the complementary DNA strand to the strand above that we see, the GACGTGCA? And so the first step here is going to be to come up with a complement. So that would be T, and then a G here, and then a C, and an A, and a C, and so forth. A sequence that basically looks like this sequence right here, or is kind of backwards of this sequence. And so, we go T, G, C, A, blah, blah, blah. And then our second question is going to be, which way should it go? Should it have the 5' on this end, or should it have the 5' on this end? So this is a complementary DNA strand, so if one strand is running from 5' to 3', the other one should be running in the opposite direction. So that means it's going to go 5', T, G, C, A, C, G, T, C. That's this answer right here, A with the 5' on one end, and then T, G, C, A, C, G, T, C. So the answer we look for here is A. Okay, so our second question is, what direction is this nucleotide in question in? Does it have its 5' to the left, or its 5' to the right? So why don't you go ahead and pause this if you want a minute to think about it, and then I'll continue. Okay, now I'm going to continue. So we learned that each nucleotide in the same strand is in the same direction. So this 5', this G here, we see has its 5' to the left, so it must have its 3' to the right, connected to the 5' of this A, and the 3' to the right, and then this C has the 5' to the left, and the 3' to the right, and so forth, all the way down the line. So that means this T would have the 5' to the left and the 3' to the right. And so the answer we're looking for here is B. All right, let's do the next question. Okay, so our question is, the pictured DNA is part of a gene. Which strand is the template strand? Go ahead and pause it if you want a minute to think about it. Okay, so here all we've done is shown you a two-stranded DNA molecule. So we actually don't know until I tell you where the promoter is or which part is the gene. We don't know which strand is the template. It could be that transcription proceeds in this direction using the left strand as a template, or it could be the opposite. It could go bottom to top using the right strand. Without more information, there's no way to know. A double-stranded DNA, we don't know which direction it will go in until we know about the promoter in which the gene is. A common answer in question was that both strands are transcribed, and we learned in class that in fact it's infrequent that both strands of a DNA encode genes, encode proteins. So generally, for a given piece of double-stranded DNA, a given gene, transcription is only going to be off of one strand. There will only be one template. So both, though possible, is not a common answer. So in this case, we don't know if it's left, we don't know if it's right. It's probably not both. And so really, it's not possible to say, so we're looking for answer E. Okay, so now we have a similar question. What about this double-stranded DNA? Is the top strand the template, or is the bottom strand the template? Go ahead and pause it if you'd like. Okay, so this is a similar case, right? We've just taken the DNA and we've rotated it. Instead of being up to down, now it's left to right, but it's still a double-stranded DNA, so we need more information to know which strand is the template. The point of this exercise is to remember that just because we're accustomed to reading from left to right, or we may have seen things depicted some way, that a double-stranded DNA is not, we don't know anything about it except for that we've chosen to show it in this way. So it's not possible to say. So here's an example. If you look, you'll see that this double-stranded DNA is exactly the same one but flipped over at 180 degrees. And so if we can take a molecule and represent it either in one way or in the other way, then it must be the case that we can't tell from what's on the left. See, the TGCA here is the same TGCA there. So we don't actually know just from looking at it which strand is the template. It's not always the bottom. It's not always the top. We need more information. Okay, so now in our next question, now we got a little bit more information. And so if this DNA to the right, if this is the gene, it's like one of these strands, this is the transcribed region, and if this is the promoter, then how can we figure out which of these two strands is the template? And so as we might remember from class, this is actually a little bit complicated, and so what I suggested is don't imagine that you can just look at this and immediately know. So instead, let's just take it step by step. So the first thing we want to know is figure out the direction of transcription. Does it go left to right, as the molecule is shown, or does it go right to left? Well, if this is the promoter specifies where transcription starts, so that means transcription starts here. And if this is the gene, then it means transcription is it's moving from the left to the right. So that means transcription is going to start here and move from the left to right. And so that gives us some information, and now we can just try out the two possibilities. Okay, so I just blew this up a little bit and separated the two strands to give us a little space. So one possibility is that the top is the template strand. If that were the case, then this is going to be the transcriptional start site at the A. Okay, so that gives us a start. And we remember that this is going to go in the opposite direction. The top strand is 5' to 3', so the template, the synthesized strand, the RNA that's being transcribed, would have to go in the opposite direction with the 5' at the right and the 3' at the left. So we know from our rules of nucleotide structure that growing nucleotide strands only grow on their 3' end. So if it were the case that we started the nucleotide here, started the strand here, then we know that the next one would have to go in towards the 3', that the next nucleotide would be added onto the 3' in this A, and so now we'd have this U with the 3' hanging off, and then we would add a C. So that's not actually the direction things have to go. Remember, we agreed that the transcription is going to proceed to the right, away from the promoter and along the gene. So that doesn't quite work. And so now we can try the other strand. So that would start with a U here. So if this is the template, then it would be a U. And now we've got the 5' of the template strand to the right and the 3' of the template strand to the left. So now this means the U is going to be the other way around, with the 5' to the left and the 3' to the right. And so now when this strand grows, when we add nucleotides, it's going to be added to the 3' end. And so that it would continue to the right. So this is exactly as we expect. We start at the promoter and proceed to the right. And so that tells us that this is only going to work if the bottom strand is the template strand. So in this case, we would be looking for the answer, the bottom. Okay, let's do one more. So if the mRNA that is synthesized from this gene goes GGGG, CU, C, GAG, UUAA, which one is the template strand? So some of you may be able to see this immediately, but I suggest that it's worth walking it through. So here we kind of have four different possibilities, right? So until we work it out, we don't know. Maybe transcription starts here with this as the template and goes to the right. That would give us this as the transcribed region. Or maybe it starts here and goes to the left. Or maybe this is the template strand and it starts here and goes to the right. Or maybe it starts here and goes to the left. So we can just try out these four possibilities and see which one of them both makes sure that we get this region transcribed and that it gives us the right sequence. So let's start with this one. So that would be, if this were the first nucleotide, now the top strand has its 5' to the left. So that means that this complementary strand is going to have its 5' to the right. So if we were to start here, what's the next nucleotide that's going to be added? Well, growing nucleotide strands only grow on their 3' end. So in fact, they would grow out in this direction, which is not what we're looking for, right? We're looking for something that would transcribe this region, not transcribe this region. Okay, so that one doesn't work. So let's try this next one. Well, what if this U were the first nucleotide? So now the bottom strand has its 5' to the right and its 3' to the left. So this first U would have its 3' to the right, the opposite way. And so now we would add nucleotides to the right. But if we actually work out the sequence, see this sequence then, if this were the transcribed region, it would start UUAA. This sequence does not start UUAA, it starts GGGG. So this is not the proper one either. Okay, so we could similarly start here with the C. So if we started with this C being complementary to this G, again the 3' is going to be the right, and so you may already see that this would lead to us extending to the right, which isn't what we want because we want to transcribe this region. And so that one doesn't work, and so we have our fourth possibility. The fourth possibility would be that we start with this G complementary to the C. Again, the 3' is going to be to the left because it's opposite the template strand. And now when we extend out, if we worked it all the way out, we would see that, yes, in fact, it is G, 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 C, U, C, G, A, G, U, U, A, A. So that would give us exactly this strand. And so we see that out of our four possibilities, this is the one that's going to work. And that's with this as a start site, but more importantly, with the top strand as the template strand. And so our answer here would be the top. Oh, one more question. And so under these circumstances, our question would be, where's the promoter? Well, what's the definition of the promoter? The promoter is the thing that's right before the transcription start site. We decided that the transcription start site is here, moving left. So that would mean that this region is the promoter. So our answer that we look for is C. Okay, thanks very much.